blood spatter experiment is a way to study how blood behaves when it hits the surface and how to understand the outcome. It helps investigators recreate what happened in crime scenes. We could change the height, the surface, or even the amount of blood dropped during an experiment. Most of the time, the higher you drop the blood, the larger the blood spatter is. By studying the distribution, size, and shape of bloodstains, investigators can gain insights into the weapon used, the direction of the attack, and even the potential sequence of events.
Blood types are determined by the presence or absence of certain antigens on red blood cells. These antigens are like tiny flags that identify your blood type. There are two main groups of antigens, the AVO group and the RH group. In the AVO group, there are 4 blood types, A, B, AB, and O. People with blood type A have A antigens on their red blood cells. People with blood type B have B antigens. People with blood type AB have both A and B antigens. And people with blood type O have neither A or B antigens. The RH group is determined by the presence or absence of the RH antigen. If you have the RH antigen, you're RH positive. If you don't have it, you're RH negative. Antibodies are also involved in blood typing. For example, people with blood type A have anti-B antibodies in their blood, while people with blood type B have anti-A antibodies. If a person receives a blood transfusion that is incompatible with their blood type, their antibodies can attack the transfused red blood cells.
Imagine a long twisted ladder. That's the basic structure of DNA. The sides of the ladder are made up of sugar and phosphate molecules. Now, let's focus on the rungs of the ladder. These are formed by pairs of nitrogenous bases. There are 4 types of nitrogenous bases: adenine, guanine, cytosine, and thymine. These are all represented by the first letter of their name, A, G, C, and T. These bases always pair up in a specific way, while adenine always pairs up with thymine and guanine always pairs up with cytosine. This is known as Ho's pairing. This specific pairing ensures that the DNA molecule is always replicated accurately, passing on genetic information from one generation to the next. A nucleotide consists of 3 parts: a sugar molecule, a phosphate group, and a nitrogenous base.
Remember, blood types also include the Rh factor, positive or negative, which is another antigen. For example, someone with type A blood and the Rh antigen will be A positive. Transcription is the process of copying the genetic information stored in DNA into a messenger molecule called mRNA, known as messenger RNA. Think of it like making a photocopy of a book. The DNA contains the genetic code that specifies the order of amino acids in a protein. The mRNA acts as a messenger, carrying this genetic information from the nucleus to the ribosomes in the cytoplasm, where the protein is actually made. How does the cell know which part of the DNA to copy? Enzymes called RNA polymerases are responsible for this. They bind to a specific DNA sequences called promoters, which signal the start of a gene. The polymerase then unwinds the DNA double helix, exposing the template strand. The other strand is called the non-template strand or coding strand. It's not used as a template for transcription, but is complementary to the template strand. So transcription is the first step in protein synthesis. It's a crucial step that ensures the genetic information is accurately transferred from the DNA to the mRNA, which will then be translated into a protein. Let's move on to translation, the second step in protein synthesis. Basically, translation, where the mRNA actually becomes a protein. It's a process of decoding the genetic information in the mRNA into a sequence of amino acids, which are they linked together to form a polypeptide chain. Translations take place at the ribosomes, which are cellular structures made of RNA and proteins. How does the ribosomes know how to read the mRNA? The mRNA is reading a group of 3 nucleotides called codons. Each codon corresponds to a specific amino acid. The ribosomes use a genetic code table to translate the codons into amino acids. The ribosomes also need the help of transfer RNA, tRNA molecules. tRNA molecules carry specific amino acids to the ribosomes and match them up with the corresponding codons on the mRNA. How does the tRNA know which amino acid to carry? each tRNA molecule has an anticodon, which is complementary to a specific codon on the mRNA. When the tRNA's anticodon matches the mRNA's codon, the tRNA delivers in its amino acid to the growing polypeptide chain. Translation is really the final step in protein synthesis. It's the step where the genetic information in the mRNA is finally released as a functional protein.
Well, you might be wondering why we're doing all these experiments. It's because we actually found a body in our classroom, and her name is Ana Garcia, and we're trying to find out what happened and how she died.